So we are on to the last lesson for this chapter, and for this section we will be discussing about other food molecules that enter the energy extracting pathways and also fermentation. Now again, hopefully at the end of this lesson you guys will be able to functionally define cellular respiration, identify the reactants and products of cellular respiration, differentiate the major chemical events of cellular respiration, and finally summarize in the form of illustration or diagram the process of cellular respiration. Okay, so far we have focused on the complete oxidation of glucose, but food also includes starch, proteins, and lipids that contribute calories to the diet. These molecules also enter energy pathways as you can see here in the picture. So basically, we do not only get energy from glucose, we also get energy from other food molecules such as proteins, starch and glycogen, and also fats. Now, the digestion of starch from potatoes, wheat, and other carbohydrate-rich food begins in the mouth and continues in the small intestine. Now, enzymes snip the long starch chains into individual glucose monomers, which generate ATP as described in this chapter. Now, another polysaccharide, glycogen, follows essentially the same path as starch. Now, so you can see here in the picture, you would see how a starch molecule or a long starch chain look like and how it actually converts to smaller or individual glucose monomers so that it can be a source of energy in the form of ATP. Now, proteins are digested into monomers which we call as amino acids, and the cell does not typically use these amino acids to produce ATP, but instead, most of them are incorporated into new proteins. When an organism depletes its immediate carbohydrate supplies, however, cells may use amino acids as an energy source. So what happens is that first, nitrogen is stripped from the amino acid and excreted often as urea. Now, the remainder of each molecule enters the energy pathways as pyruvate, acetyl-CoA, or an intermediate of the Krebs cycle, depending on the amino acid. Meanwhile, enzymes in the small intestine digest fat molecules from food into glycerol and three fatty acids, which enter the bloodstream and move into the body cell. Now, enzymes convert the glycerol to pyruvate, which then proceeds through the rest of cellular respiration as though it came directly from glucose. The fatty acids enter the mitochondria, where they are cut into many two-carbon pieces that become acetyl-CoA. Now, from here, the pathways continue as food for glucose. Now, fats contain more calories per gram than any other food molecule. After all, a single fat molecule may yield dozens of two-carbon acetyl-CoA groups for the Krebs cycle. Conversely, the body can also store excess energy from either carbohydrate or fat by doing the reverse, which is diverting acetyl-CoA away from the Krebs cycle and using the two-carbon fragments to build fat molecules. Now, these lipids are stored in fat tissue that the body can use for energy if food becomes scarce. Now, some hibernating animals use this metabolic strategy. Now, what happens during hibernation? When winter arrives and snow blankets the ground, some mammals become dormant. Now, hibernation slows the metabolism of these animals, allowing them to convert or conserve energy when food is scarce. In some hibernating squirrels, body temperature approaches freezing and the breathing rate falls to as low as a couple of breaths per minute. The reduced energy demand of hibernation does not mean that the requirement for ATP disappears completely. Aerobic respiration must continue, albeit at a greatly reduced rate. Now, the question is, where does the energy come from? Right? Now, during winter, a hibernating bear cell slowly extract energy from stored fat to maintain aerobic respiration. Other mammals, such as chipmunks, they periodically break hibernation to snack on cached food. So most of the known organisms on Earth, including humans, use aerobic cellular respiration. Nevertheless, life thrives without oxygen in waterlogged soils, deep puncture wounds, sewage treatment plants, and your own digestive tract, to name just a few places. Now, in the absence of oxygen, the microbes in these habitats generate ATP using metabolic pathways that are anaerobic, meaning they do not use oxygen. Fermentation is one such pathway. Now, in organisms that use fermentation, glycolysis still leads to ATP, two NADHs, and two molecules of pyruvate per molecule of glucose. But the NADH does not donate its electrons to an electron transport chain, nor is the pyruvate further oxidized. Instead, fermentation or in fermentation, electrons from NADH reduce to pyruvate. This process regenerates NAD+, which is essential for glycolysis to continue. But fermentation produces no additional ATP. This pathway is therefore far less efficient than respiration. Not surprisingly, fermentation is most common among organisms that live in sugar-rich environments where food is essentially unlimited. Now, many of the microbes that live in human intestines make their entire living by fermentation. One example to that is a disease-causing protease, which is an amoeba, this one, that causes a form of dysentery in humans. Others, including the bacterium Escherichia coli or E. coli, use oxygen when it is available but switch to fermentation when it is not. Most multicellular organisms, however, require too much energy to rely on fermentation exclusively. Now, of the many fermentation pathways that exist, one of the most familiar produces ethanol, which is an alcohol. Now, in alcoholic fermentation, pyruvate is converted to ethanol and carbon dioxide, while NADH is oxidized to produce NAD+. Now, alcoholic fermentation produces wine from grapes, beer from barley, and cider from apples. In lactic acid fermentation, a cell uses NADH to reduce pyruvate, but in this case, the products are NAD+ and lactic acid or its close relative, lactate. The bacterium Lactobacillus, for example, the one that you can see in the picture, ferments the lactose in milk, producing lactic acid that gives yogurt its sour taste. Now, fermentation also occurs in human cells, or human muscle cells specifically. During vigorous exercise, muscles work so strenuously that they consume their available oxygen supply. In this oxygen debt condition, the muscle cells can acquire ATP only from glycolysis. The cells use lactic acid fermentation to generate NAD+, so that glycolysis can continue. Now, lactate concentrations therefore arise. After the exercise, when the circulatory system catches up with the muscle's demand for oxygen, liver cells convert lactate back to pyruvate. Mitochondria then process the pyruvate as usual. Many people believe that lactic acid buildup causes a pH drop in muscle cells, provoking soreness a day or two after intense exercise. This idea, however, is a myth. Instead, microscopic tears in muscle tissue are thought to be the culprit responsible for delayed muscle soreness.